Hello children, welcome to BioWise with Aanchal. Now the new academic year of standard 10 has started, and with the new academic year let us begin with the new chapters. Now I will be teaching you the entire content of bio with proper practice so that you don't require any tuitions. So let us save some money and invest some time in learning biology. Coming to the first chapter, that is the cell, which is given in your syllabus as only revision. I will be taking that lesson for standard 9. If interested, you can go for that lesson. But according to standard 10 syllabus, the first chapter of the first unit, basic biology, is structure of chromosomes, cell cycle and cell division. So let us begin with the first part of the lesson, that is the structure of chromosomes. So chapter 1, structure of chromosomes. We will be talking about the cell cycle and cell division in the next video. Okay. So first, in order to understand what a chromosome is, I hope you remember a cell and the nucleus, which has got a thread-like structure present, right? This is a cell. These thread-like structures are called chromatin fibers. Now we will explore what these chromatin fibers consist of, right? Interesting. Before knowing that, there are certain words that must be clear to you. The first one is chromatin. Okay, then you have chromatids, you have chromosomes, right? So we'll understand all these basic terms. Now when you talk about chromatin, chromatin is a thin thread-like structure present in the nucleus. When the cell undergoes cell division, the chromatin fibers duplicate or multiply, and then they condense to form a chromosome. Is that clear? And then you have certain things called chromatids. Now if I say chromosomes, chromosome structure is this. Okay. This part of the chromosome is called the chromatids. The entire structure is chromosome. And these two things are called sister chromatids because they are exactly the same, you can say replica sister chromatids. And the point of attachment is called a centromere. Is this understood, the three terms? Now when you talk about the chromatin fibers, what are these chromatin fibers made up of? Chromatin fibers are made up of two components, DNA and histones. Okay. What is a DNA? DNA is a nucleic acid. DNA is a nucleic acid. And what is a histone? Histone is a protein. Is that clear? This basic information is clear to you all? So DNA is a nucleic acid. There are two types of nucleic acid we will be studying, RNA and DNA, and the histones. So generally what happens, your DNA, we will talk about the DNA structure, is a double helical structure. Okay. It forms a thread-like structure. Okay. And it goes around 8 histone proteins, 4 on the other side. It goes and winds around the histone proteins, and this structure forms a rod-like structure which is nothing but solenoid, and the solenoid then forms the chromatin fibers. Chromatin fibers condense into the chromosomes, and chromosomes are visible only under the process of cell division. Is this clear? So let us go in detail of the DNA structure. Before that, let me rub the board. So next topic that we are going to cover in this chapter is DNA. What is DNA? DNA is a very important question in your exam. So DNA is deoxyribonucleic acid. deoxyribonucleic acid. Remember the spelling. You cannot have DI, it has to be DE. I'll explain you the reason for that. Okay. Now this DNA was studied by Rosalind Franklin. Rosalind Franklin in the year 1952. And then this lady gave a X-ray crystallography of DNA molecule, on the basis of which Watson and Crick gave the model of DNA in the same year 53. In the same year 1953, and the DNA, that model was double helical structure of DNA. Okay. Now for this he prepared a research paper and he got a Nobel Prize for the Nobel Prize in the year 1962. Now Watson and Crick got the Nobel Prize. Rosalind Franklin could not get it because she didn't survive to that period of time. Okay, it's only Nobel Prize is given to the people who are alive. So Nobel Prize was given to Watson and Crick in the year 1962 for finding out the structure of DNA, which is a double helical structure of DNA. Is that clear? Now let us go deep into the DNA. Now what is DNA made up of? Okay, deoxyribonucleic acid. The name itself is telling you the story, what it is made up of. So if I say DNA, or let us start with nucleic acid. The nucleic acid has got the components deoxyribonucleic acid. So deoxyribo, that is sugar, okay, is sugar, and nitrogenous base, and a phosphate group. So for phosphate I am just writing P, circle it and write it here. Is that clear? Since there are three components, 1, 2 and 3, this is called a nucleotide. Okay, nucleotide T, 3. Okay, nucleotide 3. So what is nucleotide? Phosphate plus sugar plus base is a nucleotide. Only sugar and nitrogenous base is called a nucleoside. Second, okay. So nucleoside has got only two components, sugar and nitrogenous base, and nucleotide has got three components, phosphate, sugar and base. It is a unit of DNA. Unit of DNA. Now when we talk about the sugar, phosphate, you all know it's this PO4 3 minus is the phosphate. Let us talk about sugar. Sugar is a pentose sugar. Pentose sugar means it has got 5 carbon. Is that clear? There are two types of pentose sugar. One is present in DNA and one is present in RNA. That is ribose. Ribose sugar is present in RNA and deoxyribose is present in DNA. Is that clear? Now when I say ribose sugar is a 5 carbon sugar. Is a 5 carbon sugar. Okay. It's a 5 carbon sugar, and when you number the molecules carbon in this, the number is given with prime, because you know that DNA has got nitrogenous base also. So nitrogenous base has got the numbering as 1, 2, 3, 4, but the sugar has got the numbering as 1 prime, 2 prime, 3 prime. What happens in deoxyribose? The sugar structure is similar, right? You understand the pentose sugar, and it has got the similar structure, exactly the ribose structure, but the 2' O is missing, and it has got only 3' OH present. This 3' acts as a site of attachment of the next nucleotide, and this 5' carbon is the head of the DNA. Is that clear? Any doubt? The structure is clear. The detailed structure is not there, but for understanding how the DNA molecules are being formed, it is needed. Is that clear? The next component of the nucleic acid is the nitrogenous base. Nitrogenous base are basically of two types, purines and pyrimidines. Purines and pyrimidines. Is that clear? What are purines? You have to know the names A and G. And what are pyrimidines? T and C. Adenine, guanine, thymine and cytosine. Children, a very short note, handwritten note on this topic will be present in the description. Kindly download it and go through the same. If you have any doubt in understanding, put it in the comment section so that I can clear your doubt. So, adenine, guanine, thymine and cytosine. These are the nitrogenous base present in the DNA. But in RNA what happens? This thymine is not there. Instead, there is another nitrogenous base which is called uracil. According to the Watson and Crick DNA structure, there is a base pairing rule. That means A base pairs with T and G base pairs with C. And this base pairing you have to remember. A 2 hydrogen bonds thymine, guanine 3 hydrogen bonds cytosine. Did you understand? Adenine binds with thymine by 2 hydrogen bonds and guanine binds with cytosine by 3 hydrogen bonds. Is this clear, everyone? Yes. Any doubt, children, put it in the comment section. I will get back to you. Okay, and kindly see the description. Okay, like if you think it's worth understanding, and share and subscribe, right? That will be helpful for me too. Yes. So is this clear? Next we will talk about little bit more detailing in the structure, right? Let me rub the board first. Now we will be talking about the molecular structure of DNA and the DNA model. How you have to draw the diagram? First of all, for DNA you have to make a structure like this and follow this line and then take a turn and then this. Is that clear? And you have the ladder, this kind of structure. You need to remember nitrogenous bases, no? They form the rungs of the ladder. Okay, this is the structure of your DNA. You have, if you have to label the same, you have this one, a larger angle as major groove, this smaller angle as minor groove, right? These here, if you have A, you have T. If you have G, you have triple bond C. Again you have T, so double bond A. If you have C, then triple bond G. This is the base pairing rule, okay? And when you see the strands of DNA, if it is 5' here, remember 5' of the ribose sugar, deoxyribose sugar, so it will be anti-parallel. So this line is going to be 5' and this line is going to be 3'. So what is the basic structure of DNA? First is, it's a anti-parallel strands. The two strands of DNA are anti-parallel, right? They never meet each other. There are 10 to 12 base pairs. This is just before the turn, there are 10 to 12 base pairs. Base pairing rule, the complementary base pairing rule states that A double bonds with T and G double bonds with C. Is that clear? Now the phosphate, if you see the structure, you have the ribose sugar, right? Here you have, this is the ribose sugar, okay? Here you have got the phosphate group, okay? And here you have got the nitrogenous base, okay? So this is how your DNA looks like, the nucleotide looks like, okay? This is 1 prime, this is 2 prime, 3 prime, 4 prime and 5 prime. So this structure of phosphate, sugar and nitrogenous base is a nucleotide. Is that clear? Now these nucleotides will assemble together to form a DNA. Fine? Any doubt that you have not understood? Put it in the comment section. I hope all those who are watching my video have subscribed my channel. That is great. Thank you. Now let me rub the board. Next we'll be talking about how is this DNA packed in your cells. Now if you see the size of the nucleus, you can imagine what is the size of the nucleus. Each nucleus contains DNA and that length of the DNA is around 2 meters. And you have to fit that 2 meters DNA in a chromosome which is present in the nucleus. So there must be a proper packing done for these DNA. So let us see how the DNA is packed, and this is an important question for arrange in logical sequence, and there is a homework coming to you children after this, okay. Now when you talk about the DNA, now the DNA molecule, yes, the DNA molecule. Now this is your DNA, which is made up of what? A nucleotide. A nucleotide, smaller than nucleotide is a nucleoside. So nucleoside, you add a phosphate group to it, it becomes a nucleotide, which is nothing but PSB, that is your phosphate, sugar and base, and then it forms the DNA. The unit of DNA is a nucleotide. Then the DNA molecules becomes thread-like, and there are something called histone proteins. The name is given as H1, H2, H3, till H8. There are 8 histone proteins, 4 front, 4 behind, forming a square cube-like structure. So now this winds around the histone, and when you see this structure, there are these kind of structure, rod like structure, and the DNA is wounded around it, right? It forms a rod like structure which is called a solenoid, and then this rod like structure is going to form a chromatin fiber. This is forming a chromatin fiber which is getting coiled in the chromosome. Remember chromosome? So now this structure is going to coil itself and fit it into the chromosome, and this model is called a nucleosome model. Nucleosome model. So nucleotide is a unit of DNA, and nucleosome is a unit of chromosome. Is that clear? So in the meantime, there is a solenoid, then you have chromatin fibers fitting into the chromosomes. So you can have nucleoside, DNA, nucleosome, solenoid, chromatin fibers and chromosome. This question is arrange in logical sequence. And there is one more type of question which can be asked, that is your ratio proportion type question. Nucleotide is to DNA, so nucleosome is to chromosome. Okay, any doubt? So this is the nucleosome model. Now your question, what I was talking about, who gave the model of nucleosome, or name the scientist who has given the nucleosome model in the comment section. It's something new that you are going to research, right? Science is just not understanding the concepts. Science is also for research. So I want to know the name of the scientist who has given the nucleosome model in the comment section. Any doubt? Have you understood? So let us move to the next topic. Before that, let me clean the board. We have understood DNA. Now DNA is forming the nucleosome, nucleosome the solenoid, solenoid the chromatin fibers, and chromatin fibers finally forming chromosomes. So what are chromosomes? And what are actually, I want to discuss about the types of chromosomes. Okay, so chromosomes are of two types. What are those? Anyone? Autosomes and allosomes. Allosomes are the sex chromosomes which determines the gender. Okay, and the autosomes are the somatic chromosomes common to men and women. So when I say a male, then what do you mean? What chromosomes it has? And if I say female, then what type of chromosomes she has? So if I have to, this is a symbol for male, a circle with the arrow. The male has got 44 plus XY, and the female has got 44 plus XX. So 44 is the autosome, and XX and XY are the allosomes. Is that clear? Now this is the basic genotype of a human male and female. We have got how many chromosomes? 23 pairs. That means 46. Female has got all 23 in pairs, but male has got 22 pairs plus XY, right? And female has got 22 plus X pairs. Is that clear? Now this means that when these male and the female, I am talking about human beings, when they may form gametes, then the number of the chromosome is halved. So we have a concept of diploid. Diploid means double set of the chromosomes, 2n. For human, 2n is equal to 46. Okay, and haploid means half set of the chromosomes which is present in the gametes, denoted by n, which is 23. Is that clear? Now why I have told you this? So that it triggers your mind for us to move into the next part of the same lesson, which is cell division, cell cycle. Okay, so see you in the next video for the detailed description of cell cycle, cell division. For the important notes of this chapter, you find it in the description. Please download it and go through the same. Please like, share and subscribe my channel if you think it is worth sharing. Happy learning, children. See you again for the next video.